Good morning. My name is Dr. Lissette Otero, Chief Medical Officer and Laboratory Director of License. I apologize in advance for my English. I'm not a native speaker. If there is something you don't understand, there will be a section of questions at the end of the presentation, and we can also send you the presentation by email. Welcome to our new webinar series. In this opportunity, we will talk about telomere diagnostics and its clinical applications in the fields of inflammation, arthritis, and exercise. We will talk about how our telomere analysis technology can be useful in earlier detection of clinical risk of presenting chronic inflammation and arthritis, and also in evaluating the effect of exercise on the aging process. For those of you who may not be familiar with our company, I'd like to make a brief introduction. Lifeline was founded in 2010 as a spin-off of the Spanish National Cancer Research Center. Our headquarters are located in Madrid. Currently, we are the world leaders in the field of telomere diagnostics. We are the only Spanish laboratory fully accredited as a CLIA lab and also under the ISO 15189 standard, which accredits us to provide our services in more than 65 countries worldwide. Our TAT is the only technology in the world that's commercially and clinically validated for measuring telomeres at the individual chromosomal level. In case this is the first time you are attending our webinar or you have never worked with us before, I'll go through some of the most important aspects of telomere biology. Telomeres are structures located at the end of chromosomes formed by a repetitive sequence of no-coding DNA, which function is to protect the coding DNA, playing a very significant role in the aging process. You could consider telomeres as a biological clock. By measuring them, you can have a clear idea of how close is a cell to its senescence. Short telomeres are a contributing factor to virtually all age-related diseases. Every time a cell divides, its telomeres get shorter due to the end replication problem. This implies that every cell has a limited replicative capacity with a limited number of division cycles. This number of cycles is known as the Hayflick limit, which goes from 50 to 70 replication cycles depending on the type of tissue. After reaching this limit, cells enter into senescence and ultimately go into apoptosis. In this slide, we can see how the aging process is being driven in large part by telomere attrition. Every time cells divide, their telomeres get shorter, cell functionality decreases, and so does functionality of tissues and organs. All of this happens as part of the aging process, which leads to the occurrence of age-related diseases. The correlation between telomere shortening as a major contributing factor to age-related diseases has been widely studied and supported by more than 25,000 publications. Why should we care about telomeres? Short telomeres drive damage to the DNA into the cells. This affects tissues and organs and systems, leading ultimately to system dysfunction and the occurrence of age-related diseases like cardiovascular diseases, cancer, fertility problems, etc. In 2012, Over et al. Carried out a study where the telomeres of more than 800 individuals were measured. And as you can see in the graph, telomere length can be very different from one individual to another at any given age. Telomere attrition rate is also highly heterogeneous. You can see it in the graph. It is represented by the slope of the graph. This attrition rate can vary widely. Even if you were born with shorter telomeres than the average for your age, You can make changes that will impact your telomere attrition rate, decreasing the slope. This indicates that your aging process has slowed down, decreasing your risk of having age-related diseases. What is the ultimate goal of doing all these changes in our lifestyles? Is to maximize the health span, to live the longest in the healthiest way possible. Now, what are the main factors impacting our telomere length? Genetics certainly play a partial role in determining telomere length, and until today, we still can't do anything to change this. On the other hand, we also have the environmental factors. How you live, where you live, what you eat. They play an even larger role, and you can actually do something about them. For example, you can change and improve your lifestyle habits and ultimately improve your overall health status. The focus on today's session will be on the impact of exercise and chronic inflammation management on your patient's aging process. To start with, we will review what inflammation is, and afterward we'll talk about the correlation between telomere length, arthritis, and exercise. We will start talking about inflammation. What is inflammation? It is the body's natural defense against damaged cells, infectious agents like viruses or bacteria. It aims to remove these harmful or foreign invaders and heal itself. So, inflammation is not always bad. We must distinguish between acute inflammation and chronic inflammation. Acute inflammation starts quickly and generally disappears in a few days, while chronic inflammation can last for months or years as a result of failure to eliminate the cause or repeated exposure to the agent. Thus, chronic inflammation is not part of the body's natural healing process. It can become root cause of many diseases and accelerate the aging process of the individual. This chronic inflammation state may lead to organs and system damage and ultimately to dysfunctionality and diseases of these systems. For example, inflammation of venous and arterial walls leads to cardiovascular disease. Chronic liver inflammation leads to hepatic dysfunction and failure. Inflammation prevents the proper repairing of bones and can cause muscle weakness and pain. What are the main factors contributing to this chronic inflammation? Environmental factors such as air pollution, which promotes inflammation and contributes to insulin resistance. Stress activates the same parts of the brain as physical pain, leading to an inflammatory response. In overweighted patients, bulging fat cells send out a distress signal to your immune system. This immune system overcompensates for the intake of toxins during, for example, smoking. Also, the intake of high glycemic index foods promotes inflammation. Having an overreactive immune system triggers the release of pro-inflammatory cells and substances like cytokines that might lead to tissue damage. How does chronic inflammation impact telomere length? Chronic inflammation from recurring trauma or noxa can result in cell death, scarring, fibrosis, and loss of tissue function. In states of inflammation, subsequent increases in oxidative stress and cellular division lead to the accelerated erosion of telomeres. Due to this correlation, checking the condition and evolution of your telomeres annually becomes a key indicator to monitor how your efforts to reduce inflammation in your body are impacting your overall health and aging process. How can we reduce systemic inflammation? For example, by making changes in our lifestyle habits. You can teach your patients how to cope with the stress, how to improve their diet, avoiding inflammatory food, how to reduce their oxidative stress, avoiding exposure to toxins, quitting smoking, for example, improve their sleep, adding exercise to their daily routine, We can monitor inflammatory state of your patient by measuring erythrocyte sedimentation rate, C-reactive protein, plasma viscosity. All these are tests that are commonly used as markers of inflammation. Due to the previously exposed correlation between inflammation and telomere length, combining these tests with checking the condition and evolution of your patient's telomeres annually will help you to get a more complete picture of how your patient efforts are helping them to reduce inflammation in their body and monitoring the positive impact on their overall health and aging process. If you want to study more in depth the correlation between chronic inflammation and telomere length, we provide you with a comprehensive list of publications that you can review later. Now we will review the correlation between arthritis and telomere length and how we can use this as a biomarker to assess risk and delay early onset of the disease. What is arthritis? Arthritis is not a single disease. It's an informal way of referring to joint pain or joint disease. Common arthritis joint symptoms include pain, stiffness, swelling, redness, decreased range of motion. Also, systemic symptoms as fatigue, fever, loss of weight may be present. Symptoms can be mild, moderate, or severe. Severe arthritis, particularly if it affects your hands or arms, can make it difficult for you to do daily tasks. Arthritis of weight-bearing joints can keep you from walking comfortably or sitting up straight. In some cases, joints may become twisted and deformed. There are more than 100 different types of arthritis and related conditions affecting people of all ages, sexes, and races. We will focus on the two more prevalent ones, degenerative arthritis or osteoarthritis and inflammatory arthritis, rheumatoid arthritis. Let's talk about the main factors affecting arthritis. Carrying excess pounds put stress on your joints, particularly on knees, hips, and spine. Obese people have a higher risk of developing the disease. Some types of arthritis run in families, so you may be more likely to develop arthritis if your parents or siblings have the disorder. The risk of disease increases with age. People who have injured a joint, perhaps by, for example, playing a sport, are more likely to eventually develop arthritis in that joint. And women are more likely than men to develop rheumatoid arthritis. There is a correlation between shortened telomeres and the disease in both osteoarthritis and rheumatoid arthritis. Chronological age, as we said, is an important contributing factor in both conditions, but in the case of osteoarthritis, genetically determined factors modified by additional risk factors ranging from joint alignment to injury to activity level to obesity may play a greater role. Although it is unclear whether telomere shortening is the cause or the consequence of rheumatoid arthritis, there are some possible explanations for the association between the two. Telomere shortening results from repeated mitotic divisions, exposure to oxidative stress, or DNA repair mechanisms. Rheumatoid arthritis is associated with inflammation. There is an increased oxidative stress and leukocyte removal, which directly cause accelerated telomere attrition. Rheumatoid arthritis patients may have congenitally shorter telomeres with decreased functionality. This makes them more susceptible to having the disease. Telomere shortening can be prevented by upregulation of telomerase expression. Genetic variation in telomeres may be associated with shortened telomeres. How can we better control arthritis? Minimizing inflammation, strengthening bones, and boosting the immune system. And these are some of the more positive and effective actions an individual can take to ease the symptoms of the disease. Certain foods can help control inflammation, like those comprised by the Mediterranean diet, which emphasizes fish, vegetables, and olive oil. Also, the intake of antioxidants that have a protective effect on chondrocytes and mesenchymal stem cells against oxidative stress and inflammation induce cellular senescence and apoptosis. We should advise our patients to add exercise to their daily routine. We must consider the state of our patients in order to indicate them the type of exercise that better suits their actual health and fitness levels to avoid injuries. With autoimmune and inflammatory type of arthritis, early diagnosis and aggressive treatment is critical. Slowing disease activity can help minimize or even prevent permanent joint damage. The goal of treatment is to reduce pain, improve function, and prevent further joint damage. Here you have a list of publications of papers studying the correlation between telomere length and arthritis in case you want to review them. Now we will talk about how we can impact the aging process through physical activity. The chart 2 points out towards multiple evidence correlating exercise with telomere length maintenance. Excuse me. Like in this paper, where researchers found that women and men who were less physically active in their leisure time had a shorter leukocyte telomere length. Exercise helps to maintain leukocyte telomere length, and maintaining telomere length leads to maximizing health span. Another research project conducted on almost 6,000 adults by Brigham Young University in 2017 revealed that regular physical activity helps to slow down telomere attrition. The more physically active an individual is, the less biological and cellular aging takes place in the body. The right amount of daily exercise can lead to a more healthy aging. The level of physical activity not only maximizes your heart's pumping power, but it also helps with muscle growth, as well as the production of anti-aging hormones such as human growth hormones and DHEA. However, it is also important to mention that extreme exercise, on the other hand, can promote inflammation, as it promotes the production of free radicals, leading to inflammation. As a physician, you may implement TAT in your clinical practice and present telomere tests as a call to action for lifestyle changes and health therapy adoptions. Again, we have here a list of relevant papers studying the correlation between maintenance of telomere length and exercise. From here, we will describe how to implement our TAT test in your clinical practice. Our TAT should be considered a risk stratification biomarker. Even when a single measurement gives us valuable information about where do our patients stand at the moment, the real value of the test comes from making several measurements. This will produce longitudinal data, allowing us to know how a patient is evolving, how the personalized program that you have designed for him or her is impacting on their telomere attrition rate, if all these interventions aim at controlling chronic inflammation, like exercise, for example, are working, or if you need to modify the program or add different actions. What we recommend is to do the TAT prior to the beginning of the program to get a baseline, and then retest your patient after 1 year. Repeated on an annual basis, it is like a report card of whether lifestyle programs are working or not, indicating whether your patients are aging at a normal pace and at an accelerated pace, or ideally at a slower pace due to the efforts that they make to take care of themselves. like working out, eating right, managing stress, etc. Depending on result, we can adjust and take measures if we see that this program is not working. This is a results report example. Here you can see in the graph the result, the black point is the result of your patient, and you can compare it with other people that is the same age of your patient. We also give you the 20th percentile, the percentage of short telomeres compared with the control database. Our test is set up to be longitudinal. So if your patient has done the test more than once, all the results are shown in the report, like you can see in the graphic. People have the opportunity to complete an anonymous health questionnaire online. If the patient reports to have any of the protective or risk factors that impact telomere length, these factors will appear highlighted in blue in the report. This slide shows the evolution over time of different technologies developed to measure telomere length. Our TAT is the most recent technologically advanced, clinically validated, that allow us to obtain not only the average telomere length, but also many other parameters like the median telomere length, the percentage of short telomeres, which provides clinical relevant information about where your patients stand in their aging process and the likelihood of presenting age-related diseases. No other technique can provide neither all of this information nor with such accuracy as our TAT can. When compared to the other two most commonly used methods for telomere measurement, these are qPCR and flowfish, TAT is the only one that meets all three characteristics required for being a clinically useful test. Scalability. This allows us to process a great number of samples at the same time. This decreases the unit cost of the test, making it affordable for the patient. Total reliability, as our test is reproducible and accurate, and this is granted by our strict quality control program. And also the clinical value we have talked about before, that only a clinically validated test can provide. This graphic summarizes all these attributes of our test. It allows multiple sample processing. We can obtain median telomere length and percentage of short telomeres from single cells, not only from populations of cells. Just to summarize and to finish, TAT is a cutting-edge diagnostic test that looks at your aging process at the cellular level. Possibly the best overall biomarker of health that exists. It is a simple test. Not special preparation or fasting for blood collection is required. Only one tube with 10 millimeters of blood is needed. We strongly recommend patients to complete the online questionnaire, as this will facilitate the interpretation of the test results. The results report will be delivered to the physician in approximately 2 to 3 weeks. This depends on the country of origin and shipping time for samples. We can provide this report in several languages. Now we'll make a short break to allow a few minutes for people to ask any questions. I hope you found this presentation useful and helpful. Thank you very much for your attention.